Ballard's boat.
And then, what, didn't we have a tub one today?
Yeah. I would do anything for tub.
Oh, yeah, for the tub of
I
tea.
would do anything for tub. We didn't do anything for tub. We just made a song about it.
Yeah, it took me a while to find out that there were Nautilus albums.
Yeah, yeah.
I didn't know. So many musicians on board.
Yes, indeed. And now this area that we are going to, the seamount north of Jarvis Island, is a never before explored seamount. So we're very excited to check it out, see what we might find. As I said yesterday, we really saw some interesting and exciting life and geological formations. The flows yesterday were really cool. So I am very excited to see what we find when we get down to the bottom. And I'm also very excited that you all get to join us. So again, you can send in your questions to nautiluslive.org in the white box below the video feed. And again, if you want to do some math at home and figure out how long it's going to take for us to get to the bottom, we are descending 1,800 meters at a rate of 27 meters per minute.
And we have gone 747 meters.
Ah, here we go.
And if you don't like math, I'll tell you the answer in 2 minutes.
We were talking about that the other day, weren't we? How, you know, once you find a way to apply math in a way that actually relates to things that you're interested in, things that you do, it becomes a lot clearer the importance of math.
Because at that point, it's a tool.
Yeah.
You know, it has an application.
Are those pink wiggly things we're seeing fish or worms of some kind?
I was just looking at that. No, it was much larger than a krill. I was just looking at it and wondering what it was as well. I'm not sure.
I've seen a few of them.
Yeah. One of them looked like it was part pink and part white. Did you see that one?
Yes, I did.
Yeah.
Yeah, even though we're just going through the blue water right now, you can see quite a bit if you watch the screen. There's some jellies that have floated by, some more of the free-floating sea cucumbers that we've been seeing, some fish, squid, snothouses, all the best things. Ooh, I've got a question about marine snow here. Would we tend to see more marine snow closer to land masses?
There are certain regions of the ocean that have more concentrated marine snow. So areas with greater upwelling, which often occurs closer to shore, would have more marine snow. Out in the larger pelagic ocean far from shore, there is frequently a lower density of marine life, so that would also cause less marine snow.
Mm-hmm. Because again, for viewers at home, the marine snow is debris falling through the water column. So, you know, pieces of those snothouses, scales, skin cells, all that sort of stuff, gametes and debris from poop and things getting eaten but not all the way eaten and all that sort of stuff. So the ocean is a soup. Oh, hello to our viewers from the Sand-Soaky Public School in Sydney, Australia. We've got a class of 6- and 7-year-olds watching our dive today. So thank you so much for tuning in. Feel free to send any questions that you might have in particular, but it looks like you are wondering what we're up to. We are currently actually moving down through the water column to the seafloor. We just got started on this dive. We're going about 1,800 meters below the surface of the ocean to explore this area. It's something called a seamount. So it's a mountain that's underwater, and we're going to look at it to find out what lives here. We're also going to be looking at the rocks here so that geologists can learn more about the area, like how it formed, when it formed, and what kind of changes may have taken place over time. So we're going to be in the blue water for a little while, but, you know, keep your eye out and see what you can see as we descend, what types of life you can see. And once we get to the seafloor, we'll be exploring that seamount. But again, thank you to 1N from Sandstokey Public School in Sydney, Australia. Really glad to have you watching today. Ooh, what are the things underwater that have an illusion of water or little lakes? I think you might be thinking of brine pools on the seafloor. So that's super salty water that's being released from the seafloor and sort of forms a pool and does look like a lake. And there's some awesome videos on our website of brine pools that Nautilus has come across before. So if you go to nautiluslive.org and check out our photos and videos tab, you can check that out and see those brine pools in action.
Okay, so I lost it a little bit on that time. It's been about 6 minutes, but I have the answer.
Give it to us, Chris.
As of now, we should be on the bottom in just over half an hour, 33.3 minutes.
Awesome. Thank you. So it seems like that the official launch watch is still going to get a bit of the blue water when they relieve us for dinner.
Yes, they will.
Valet service, they'll get it right down just in time for us.
Perfect. And no apologies necessary for our viewers who were confusing me with Allison yesterday. It is a wonderful confusion. I am honored. But we are different people.
I also respond to Al, by the way.
Oh, do you?
Yeah, my family calls me Al.
Oh, that's funny. We will become one. Oh, we got another question from our class of 6- and 7-year-olds watching, wondering how long it's going to take us to get down to the seafloor. And our navigator, Chris, just gave us an estimate of about half an hour until we get down to the seafloor today. So we'll be moving through this blue water, and then we'll be exploring on the seafloor in about half an hour.
What time of day is it in Sydney?
I do not know off the top of my head. Let me see.
Daytime.
I know where we are. It is the 4th of July, and it is 4:48 p.m.
It is about 1 p.m. for them on July 5th. So middle of their day.
What's it like in the future?
Did I find any cool rocks?
Yeah, we're not only just interested in biology here, we also love our rocks on nautiluslive.org. Were you happy with the rock samples you collected yesterday, Kevin?
Yeah, they're pretty good. I won't know for sure until we cut them open because a lot of them are encrusted, so you can't really see them.
Do you have to wait for them to dry to cut them open?
No, but we don't really have a saw that works well on the board right now. So I'm going to let the good folks of UOI, University of Rhode Island, do all the work for me. And I'll send them a lovely sample request.
Beautiful. Yeah, because all the geology samples go to Rhode Island, and all of the biology samples go to the Museum of Comparative Zoology at Harvard. And they can be requested by scientists from all around the world.
Kevin, which type of saw would you typically cut a rock in half with?
A tile saw. So I use a nice saw with a diamond crusted edge. So I have different sized tile saws that work with some are massive and some are smaller, depending on what kind of level of precision I want.
They're so much fun.
What's the biggest rock that you've like cut open with a saw? Like I just imagine you with a boulder and like a huge wielding
Like
a huge
like
diamond
every day
saw.
encrusted
I pull up the Atlas
chainsaw.
stones.
Yeah, exactly.
I don't know. The biggest one I probably cut with a saw might be a bit bigger than a basketball.
Pretty
I had to cut
respectable
one side
rock.
and flip it, and then cut the other side and flip it. Things I probably shouldn't be doing with the saw, but I got it to work.
Make it work. That's what a lot of science and especially engineering is all about. Using what you got to get the job done. And if you want an idea of how far out in the ocean we are, take a look at Google Earth and you can check out Jarvis Island and see how far we are, because we have been trucking it through the ocean, through a lot of ocean.
It's what we call cutting holes in the water.
Cutting holes in the water.
Yeah.
I like that.
I've never heard that before.
Do you know where that phrase originated?
Nope. Just know that we use it. I guess for us it's moving with a purpose, but usually it's for just, you know, when you're just out there to go somewhere and you're just going fast
Just
and
booking it.
displacing a lot of water.
Very cool. New phrase. Oh, and hello to our friend Diana, it's 3:38 a.m. in the northwest of the UK. Says she's loving every minute of these dives and her coffee intake has increased to keep up with us.
I support that.
Oh, and to our friends in Sydney, they have a school assembly in a little bit, but they're going to be back watching in a while. And once you guys get back in about an hour and a half, we will be at the seafloor. So excited to have you come back and see what we find live with us here on Nautilus Live. And thank you to your teacher for watching with you. We're really excited to have you all exploring with us.
Good on ya.
We've got a question about how old the oldest coral we have ever found is. I cannot speak directly for Nautilus about the oldest coral, but I know that one of the oldest corals ever sampled and dated was a black coral that was dated to 4,000 years old about. Does that sound right, Jamie?
Yeah, older than 4,000 years.
Wow.
Mm-hmm.
Do we know what the oldest corals that we've come across are on Nautilus?
I'm not sure. Usually age doesn't get determined until fellow scientists on shore take the samples and bring them back to their own labs and investigate them further there.
We saw some, Steve might have a good idea. When we were in the Gulf of Mexico with his lab, actually, Dr. Eric Cortez, we saw some very large corals in the Gulf.
Well, then we will investigate with Steve over
like all coral questions, we will ask Steve.
Yeah, over the dinner break and get back to you soon.
I know on some of our dives last year, when we were diving in Papahana, Mokoakia, Chris Kelly and some of the scientists ashore had given, visual estimates of what we were seeing that could have been at least 1,000 years old, just based on the amount of growth that we were seeing of these corals. But again, they can't be fully dated unless they are collected and studied by scientists.
Take a quick look at them now, but usually just take them on the bottom.
Oh, and it looks like we've got another school tuning in, Kadena Memorial School. Thank you so much.
No harm.
We're excited to see any questions that you have during this dive.
Welcome. Do we know where that school is located?
I'm checking it out. Looks like Kadena Memorial School is also located in Australia. Correct me if I'm wrong, my friends at Kadena. But thank you so much for joining us today. We're really excited to have so many students watching with us on this dive.
I don't think so. I'd see about it on the
So
dinner break.
when we say a coral's over
From the
4,000 years old and they're colonial animals, does that mean that as a colony it's over 4,000 years old?
Yes.
Not one
So
individual.
like once the start of that colony,
Right.
When it began. Yeah.
There wouldn't be any individual
I'm not entirely sure how old
that lives that long.
the polyps can
Yeah.
live. Again, we can ask Coral Steve for more specifics about that.
Can't wait.
Yeah, that's a good question. Because again, corals that we're referring to, the deep sea corals and the coral reefs that you see in shallower waters are colonial animals that grow these large skeletons that build the reefs in shallower waters and build these sort of big fan-like shapes that we've been seeing in the deep waters. And it's little polyps that are eating food out of the seawater. They're eating that marine snow. A lot of the animals that we come across on the seafloor exist because they're eating that marine snow. Sea cucumbers and urchins, things like that, are all extracting nutrients and, sort of cleaning things around there. And yes, Kadena School is located in South Australia. So awesome to have so many classrooms watching today, so many Australian classrooms checking in with us. And again, we welcome your questions. We'll be going through the blue water for a little bit longer now, so let us know if you have any specific questions. If we don't get to the question right away, we will try and get back to it. We will be doing our dinner relief for a little while, so there will be a little shuffling in the control van as we all get our meal in. But again, thank you all for watching, and we are exploring a never-before-explored seamount north of Jarvis Island, which is here in the Pacific Remote Islands Marine National Monument. And if you've watched any of the other dives, you may have noticed that the corals that we see are on rocky substrate, which means rocky bottom. And so that's where the base can attach, and then that colony can start to grow. There are relatives of the corals that we see called sea pens, and some of them are able to stick to rocks, but a lot of them prefer to be sort of rooted into the sediment. So some different animals prefer different areas for growth, and the corals that we were seeing definitely prefer those rocky areas. And we'll also tend to find more corals and larger corals where we have more of that nutrient flow, so the marine snow moving through the area for them to catch with the polyps and to consume.
What do you call it when a coral writes a letter to its cousin?
I don't know, Steve. What do you call it?
Sea pen pals.
Adorable. We're not quite sure what's on the dinner menu yet. We'll be checking that out as we take our dinner break. And barnacles are actually, we can see them at shallow depths. We can also see them at deeper depths. And they are actually related to crabs and lobsters. They are non-mobile, or another word we can use is sessile, meaning they stick to a spot and they grow from there. They also grow a little shell, which is usually what you see is that barnacle shell. You don't usually see the animal itself. It's kind of closed up inside unless it's feeding or mating. And they do tend to encrust a lot of different surfaces. So you see a lot of barnacles all in one area. Again, because they are sessile, they stick to one place and grow. And we do see them in the deep sea, as well as in shallower depths. Now, in between these dives, we are typically doing sonar mapping, which is when we're sending a sound down from the ship to the seafloor. It's bouncing back, and based upon the way that it returns to the ship, our mappers and navigators interpret these signals to understand what is below the surface, what the seafloor might look like. Can also give us an idea of the hardness or softness of the area. And once we have these areas mapped, that's when our science team will take a look at the maps and determine the best places to dive based on the objectives, whether it's to find certain pieces of geology, for rock collection and understanding the biological communities. Our science team will use their expertise to come up with the best track to follow for these dives. So once we get down to the bottom, that'll be the beginning of our track, and throughout the dive, we follow along that predetermined track to explore this never-before-explored seamount.
You know, Al, I find it impressive that you not only know your job, but you also know, like, everyone else's job. Like, you just described my job.
That's my job.
Yeah.
I appreciate that, Chris.
Describes my job. Oh, yeah, I do that. All right.
I'm glad I do it justice. I'm also just an uber nerd and know all of it anyway. And speaking of Chris's job, the ship, the ROVs, all work in tandem together to move throughout the seafloor. So our ROV pilots are communicating with our navigators, who are communicating with the bridge of the vessel to move it to allow us to explore these tracks along the seafloor. Hello, Wayne.
Hello, Al. How you doing?
I'm doing well. Nice, relaxing blue water to start the watch. You excited? You still got a little bit of it? Oh,
Yeah.
wait. Emily's usually the one getting the blue water, right?
Yeah.
Giving her a little reprieve.
I've been fortunate. Most of my shifts have been 100% on station.
Very nice. Very nice.
So can you shut the light off and bring it back on? Just the flicker. I think if you shut the light off and then back on, that usually, yeah, that clears it up. No, no, it's the reflection off of the high reflective yellow bits.
It would not. Kadena School from Australia has a question here. Are we expecting to see any invertebrates? For example, the strawberry hermit crab. I know strawberry hermit crabs can be found on some of these remote islands. I'm not sure how deep we can find those particular hermit crabs, but we did see hermit crabs last night on our dives. When we zoomed in on some of the shots, there were little white snail shells that were being lived in by some hermit crabs on the seafloor. We also saw some invertebrates like actual snails, which were still living in their shells that they create. And a lot of the animals that we see down here are invertebrates. So things like the coral that I'm talking about is a colony of little invertebrates, meaning that they don't have a spine. And we also saw things like urchins and sea stars and jellies and sea cucumbers, all of which are invertebrates. And we do expect to see similar life when we get down to the seafloor today. So definitely keep your eyes out. We'll be seeing some really interesting invertebrates, I'm sure. Thank you for that question, Kadena Memorial School. And the corals that we see on the seafloor do grow slowly. A lot of life down on the on the deep sea, due to the really cold temperatures, high pressures, are going to be growing more slowly than you may imagine their relatives in shallower waters who are using sunlight may grow. But it also depends on the corals themselves. Some corals can grow millimeters to centimeters per year, and some are just very slow, very small, millimeters per year. So it does depend on the coral, but they are slower growing in the deep water. Jamie, are you still back there?
I am.
We've got a question about how we avoid introducing bacteria to the samples that we collect.
That's a great question. So after every dive, the vehicle gets thoroughly rinsed off. So that's a major asset. And then another way is if there does happen to be a bacteria from the surface on the vehicle, it's pretty unlikely that it's bacteria that could survive at depth. It's likely to be bacteria that's acclimated to surface waters or air, and not intense pressure and cold temperatures like we experience at depth. So the probability that after all the rinsing and the small likelihood that a bacteria from the surface, if it was there, could survive down there means there's a low probability of introducing something harmful that would survive down there when we did a sample.
Awesome. Thank you for that. And then the processing that you do afterwards is some of the ways that you process are like when you're preserving the sample and things, is that also to kind of stop any bacteria from getting onto the samples?
Yeah, I mean, we do want to keep the sample genetics fairly distinct so that when analyses are done later, we're looking at what the actual organism was rather than additional contaminants that might be in the air or something like that. So we try to minimize the amount of time the sample spends sitting around and be efficient when we're moving the sample from the water into the preservative we use, which is typically ethanol.
Awesome. Thank you for that, Jamie. And again, you're going to hear some shuffling over SPL as we get relieved for dinner, and we'll be on the seafloor soon.
Thank goodness for dinner. Didn't think I was going to get to do this.
What was that?
Do this?
Yeah, I thought I was going to have to miss the blue water.
You know we can't deprive you of this excitement.
Yeah.
I wouldn't say I'm missing it, Bob.
There we go. Only 9 minutes.
Uh-oh.
Wow, this is snowy. This is a good sign.
So I heard we were going down to 1,776 feet. Is that right?
I tried to request that, but we will indeed hit that depth as we, well, both on the way down in the water column, but as we climb up the seafloor. But we're going down to an expected depth of around 1,800 meters.
Did we bring a flag to plant at that depth?
I think Jarvis is already US easy anyway.
thinking about it now, we could have sent them down with a welding rod and a blowtorch and turned all the lights off. Have them down there with a...
Maybe we'll see some firework coral.
Hey, it looks like some viewers are curious. Do corals in deep water have different structures or physiological strategies to withstand higher water pressure? Steve?
Yeah, the short answer is not as much as you might think. Corals don't have air-filled spaces in their body, so they're completely supported by a liquid water hydrostatic skeleton. However, there is some evidence that maybe pressure at extreme depths, abyssal or hadal depths, may have an influence on how proteins fold within the tissues. And if proteins aren't folding properly, you're not going to get the right things being created. Not going to get the right enzymes being formed, things like that. So it's important to understand the effects of pressure, but it's not really clear when you get beyond the depths that we're working at.
You can increase your wind speed a little bit there.
Generally, the temperature is the thing that will harm corals, the temperature difference. Oftentimes when we bring them into the lab, just from moving them from the bio boxes, for example, to the refrigerator, some of the polyps will have small-scale necrosis occurring just over those few minutes. So we have to try and be deliberate and quick in how we process them.
Sorry? Yeah. Here, I'll slow up a little bit actually anyway. Roger. Get this landed for them pretty much, and then we'll go to dinner.
Unwarranted door slam.
Someone's very excited.
I think you should rock it right down to the seabed, Jess, and look around and let the on-watch people do all the weight balance and
I'll get them set up.
all the setup stuff.
I'll get them set up so I can do the white balance. But we'll still all stop around 50 meters.
Roger.
We had issues with the white balance last time, but right now the controllers are behaving, so I don't think we'll have any trouble.
Sure thing.
So why can't you do a white balance on the fly? You have to be static at the depth?
Well, it's best to do the white balance in the environment that you will be filming. So when we reach the bottom, that's going to be, how should I say, the portion of the dive that has the most light absorption. So white balancing at the deepest part.
Theoretically, after the photic zone, you should be fine, but
yeah,
seems to be protocol to just do it at the bottom.
Yeah. There's
yeah, so Sorry, in my reptile brain, dark is dark. Deep is deep.
Deep is deep. Yeah, the percentage difference of doing it here and then doing it at the bottom is negligible.
I thought you did it on the bottom to get some ambient, like from the reflection of the vehicle's lights off the seabed or something.
No.
It changes if you're on a sandy bottom versus a rocky bottom.
Not so much. It's the what you're testing is you're testing the water visibility
meters.
Between the camera lens and the white thing that you're testing.
Roger.
That's it. You're just
so it could
calibrating.
be particulate in the water that.
Yeah,
if there's starting to slow up.
Different sediments or different snow or.
I'll buy that for $1, Wayne.
So.
All right, I'm gonna zero my Z-bias here and makes me an all stop.
I'll stop on the winch.
Roger. You mind closing that out for a second, just so I can see where we're at? Thank you. I'm gonna change my heading around a little bit, and then I'll come down and we'll do the same old, same old. Okay, coming down.
We have a visitor.
Would you mind giving me some bubble there? Bubbly
Continuing
action.
down.
Oh, sorry, just the bubble. Yeah.
She's got it there.
I'm gonna start backing up towards you. And when we reach a 20 delta, you can hold off there for a minute if you want.
Copy, I'll stop.
All right, go ahead and see if you can turn around to face me.
Do you have a preference on the rotation?
It looks like they're going to be going more north. So if you go spinning that way, so that'd be negative 10 degrees.
Anti-clockwise is the...
Anti-clockwise is a good way to put it. Then that should set them up for when they're going up the hill.
As they say, south of the equator.
To port.
Yeah, thinking with my hands right now. How's that
Hello.
mic?
So I did an experiment. I left the, we energized the craft and got the bouncy out of the way.
Yeah, I saw that. I forgot to do that.
And we left it hydraulics onto it. And I was watching the gauges there. They didn't move at all. So, and the GFD went from 7.3 to 6.7, so maybe that helps keep it happy.
That's 6.7, that's Argus.
Or sorry, 11.1.
So what are you saying it did? It popped down to that?
Oh, my theory is if it has pressure in the linear actuator, you know, smell what I'm stepping in.
Yep.
I think the next step is the white balance. Is that correct? Roger. Paying out.
We're close enough? Yep. Okay.
Turkey.
Yeah, we're like,
Yeah,
but it's in the radar.
30 meters away.
So
Sonar, you mean?
the sonar.
Can anyone do the sonar sound effect? Go for it, Wayne. You
No.
want to?
Yeah.
I think it's more of a pew.
Burner, burner.
There's
we were
the
doing.
Yeah,
bottom.
Roger.
In sight.
Oh,
Okay. Now we see the bottom.
Land ho.
excuse me.
Land ho.
What scale do you want? 30 meters? That altimeter, I got it. I'm not deep enough. I got a uptick 50, you'll get it. So I'm still 50 off. Coming down. Is it you or Trevor that turns this one off? You don't like that noise. It's only noise. I'm winning the race. It's been about 150-ish. Pain out. When in doubt, pay out. $10 meters. I'll stop on the winch at 25 meters altitude.
Yes. Once we get some good fixes here, I'm gonna get a good DVL reset for you.
No, it looks clear.
We're old school, Jess. We're old school. We kind of like flying the ROV. Yeah. Yes.
In just a sec. Auto's off. Alrighty.
Okay.
And
It is nice to have.
reset.
If you need a bio brake, you just hit auto XY. We do. We do. It's hot. Yeah, it's all hot, hot. It's hot, hot, hot.
Nav, did you just complete the DVL reset?
Yes, we did.
Got it, thanks.
So yeah, the weight balance here, to me, seems like the right place to do it because you get some backscatter from the seabed.
Yeah. So it remembers the white balance from our last dive, and you can see the colors of the tape, the red, the green, and blue. This should
be bang on.
Or, well, it's very close because we were diving under very similar lighting conditions. So, okay. Okay, prepare for.
Yeah, that's how I've been holding it too, just to avoid the.
Okay, going for black balance. Screen will go dark.
Just to avoid the change in it. Yeah, I do too.
Okay, going for white balance.
It's not that bad that way. It's not as comfortable as the other way, but. We are old, Mike.
Okay, white balance is complete. Yeah, and I can generally notice a little bit of change in the blue tape every time we white balance.
I'll hang out for a while.
So,
I'm happy to hang out for a while.
All right.
Yeah, you can go eat. I'll eat. I hang out here and heckle with Mike for a while. The old boys in the front row. I'll show you kids how we run this ROV.
Okay, the ROV is ready.
Finally.
Finally.
I am all set.
Yeah.
Alrighty. You guys want to do steps or?
Yeah. So I
line?
put in waypoint 1, so we can go up toward the ridge, towards waypoint 2, because I want to grab some rocks on the side of this ridge. So
Roger
I think
that.
steps are probably the best for now.
Roger that.
Steps.
Kevin wants to grab rocks?
Bridge nav.
I know.
Request 20-meter step bearing 340.
340.
Thank you.
Which, yeah.
Can you make it any smaller? It's not small enough. Yeah, it needs to be smaller.
And hello to our friends on nautiluslive.org. If you haven't already seen it, we are on the bottom. We have made it to the bottom for this dive north of Jarvis Island, where we will be exploring a seamount.
Kylie, can you take a 340-degree heading?
And our regular reminder for our viewers at home that there are a couple different ways that you can watch this dive. We've got our Herc cam, our Argus cam, and our quad view that we will put up different things in that third screen, like the HIPAC map, which our navigator is using, which shows us the ship's position and the vehicle's relative positions. We also put up the sunset sometimes, so might be seeing that a little later. We'll see. But you can check out all those different views throughout the dive to get some different perspectives on what we are doing down here. And this area in 2015 actually experienced some major coral die-off close to Jarvis Island. About 98% of the healthy coral communities in this area were bleached due to an El Nino in 2015. So we're really interested in seeing the deep water coral communities around this area, as well as we get shallower, what we might see as well. So should be a pretty interesting dive here.
Can we zoom on that stalk?
I think it's a halosaur.
Oh, is it a fish?
There's a fish.
Oh, the fish that looks like a stalk then?
Zoom in, please.
Like fish.
Coming in.
Does indeed look like a halosaur.
Yes, it does.
Name meaning salty lizard. Or salt lizard. Either one.
Coming wider a little bit.
Now it looks like the sediment in this area seems to have sort of a crusty appearance over the top. Do we know why that might be?
Full wide.
Not really. Might be thicker chunks of like stony coral that have tumbled down the hill, or I don't know. But yeah, it's not as soft and nice as the other areas.
And it did look like there was also possibly a xenophyophore somewhere on that bottom there. And again, we do have the Mizo GoPro on that front porch there. You may notice it's in a different position than it's been in the past. Trying out a new position to see if it can get some good shots, also keep the camera safe.